Hello, my name is Matthew, and today I'll be talking about this paper called Melatonin Delays Leaf Senescence of Post-Harvest Chinese Flowering Cabbage Through ROS Homeostasis, as presented by Tan and Associates. This is a paper published in October 2020, and I'm presenting this here. So this paper, if you want to simplify the name, this is to say melatonin makes choy-sum leaves stay green longer because it handles stress better. All right, so what did these folks know going into this research project? Well, this is our Chinese flowering cabbage. It's a Brassica rapa, subspecies Parachinensis. Has these nice deep green glossy leaves that look quite tasty. And it flowers, and so you cut this and you send it to the supermarket, and then you sell it and people eat it. So here's our little Chinese flowering cabbage. But then there's a thing that happens where once you harvest it, there's a thing called post-harvest leaf senescence, which leads to degradation of chloroplasts. So the chloroplasts are what make the leaves green, and because the chloroplasts are degrading, this plant turns kind of yellowish. And this reduces consumer acceptance of the product. Now, what can we do here? And the question is, how can we delay this leaf senescence so that we don't get it turning yellow and we can keep it green? So leaf senescence is a form of programmed cell death. What this means is we have decreased photosynthesis, we have a remobilization of nutrients, and there's an increase in these genes called senescence-associated genes. So leaf senescence is a controlled mechanism in plants where a variety of things happen, and there's specific genes that are turned on to help regulate this process. So the initiation of leaf senescence is closely related to the reactive oxygen species homeostasis. So reactive oxygen species is also called an ROS. So I might use that term in this presentation instead of saying the whole thing out. So some examples of this reactive oxygen species are these hydrogen peroxide. So they have these free pairs of electrons that are able to do some reactions. And so this can be deleterious and problematic if you are trying to survive and not be destroyed by the inside chemistry. The next one is your hydroxyl radicals. So we have this reactive little oxygen here. It can do some interesting damaging chemistry. Then we have superoxide. So these are the three main ROS that we are going to be looking at here today. And they're often the most prevalent as well in terms of ROS stress in the plant. So what we can see is that ROS concentration is regulated by antioxidants. So ROS, they are basically oxidants, so they oxidize, and then antioxidants stop them. So there's two types. There's enzymes, which include superoxide dismutase, or SOD, catalase, the CAT, peroxidase, POD. We also have the glutathione reductase, and then the ascorbate peroxidase. These bottom two, GR and APX, these are called secondary antioxidant enzymes because they function to help reduce the second type, which are called metabolite antioxidants. So when we look at these, we see that there's glutathione. So this enzyme, glutathione reductase, helps maintain glutathione as a metabolite that can antioxidize. We have polyphenols like anthocyanidins, carotenoids like beta-carotene, which makes carrots orange. The vitamins, so vitamin A, vitamin C, like ascorbic acid, vitamin E, and vitamin K. And then minerals as well can act as antioxidants, like zinc and selenium. So let's take a look at this. If we have on our y-axis, it's just a relative unit. So this is just some kind of arbitrary scale. But on our x-axis, we have a young leaf, and then we have a mature leaf and a senescent leaf. So this is basically looking at a leaf over time. So this first line that kind of goes up and increases over time, that's our reactive oxygen species. Our second line here that goes up and then dips once it starts to senesce is our APX. This is an enzyme, the antioxidant enzyme. And then our third line here that follows a very similar pattern to APX is our SOD, the superoxide dismutase. And so what we can see from this is that there is a correlation between the concentration or relative units of these two enzymatic antioxidants with the relationship of ROS concentration. So this is called ROS homeostasis. Now, once we hit the senescent leaf area, these antioxidants decrease. And this is where the homeostasis changes. So let's look. We have our ROS here. And what happens is it promotes the production of these enzymes that we talked about. Now, these enzymes can then quench the ROS and stop them from doing things that are bad. But the ROS also turns on melatonin biosynthesis genes. Now, these biosynthesis genes make melatonin, which in and of itself is an antioxidant and can quench the ROS. But melatonin also has been shown to increase the production of the antioxidant enzymes. And so this is kind of a simple diagram of how melatonin fits into ROS homeostasis. This is adapted from a 2018 paper. It's kind of a new area of research to an extent. Now, if we look at melatonin here, this can be transported in the xylem. And there's a receptor that has been identified called CAND2 or PMTR1. And then this leads to activation of responses, allegedly, at the time of publication. So is this the newest plant hormone, melatonin? Well, in the 1970s, it was identified in coffee as potentially a mistake of roasting. And they said, oh, well, this is just in there because something about roasting the beans. But then they discovered that, oh, it was actually in these plants in 1995. And then in 2018, they believed to discover the receptor for melatonin. Now, if this is a plant hormone, we would expect to see a signal transduction pathway, different activation of genes, transportation or translocation throughout the plant, and so forth. And so we've seen some of these. So maybe this is some kind of plant hormone. Maybe this is just another molecule. Anyway, so if we look at the popularity of melatonin research in plants, so this is the number of articles published on melatonin in plants, we see that around 2013, 2014, 2015, it starts to pick up. And now we have this sharp increase in the number of publications per year. This was extracted from a 2020 article where they evaluated this. And so in 2019, one research team with a first author named Tan, they did an experiment where they applied melatonin from the outside onto these leaves of that Chinese flowering cabbage, and they found that this delayed leaf senescence. So on the top is our control. So this is no application of melatonin. Exogenous just means applied on the outside. The bottom one is the melatonin, the exogenous melatonin applied at the beginning. So at day 0, they're the same. At day 1, they're the same. At day 3, just from our own visual cue, this appears to be a bit more yellow, which we would relate to a degradation of chloroplasts. And at day 5, this is a lot more stark. Now we see down in the melatonin treatment that they're also turning yellow, but not as quickly as day 5 in control. And so now we can look at the rate. So this is a ratio, essentially looking at the amount of activity in photosystem 2. So this is deep blue here means high rate of photosynthesis. And then our kind of orangish, reddish, that is a low rate of photosynthesis. This correlates to the chloroplast degradation. So if you don't have chloroplasts, you cannot photosynthesize. And so then you'll get this kind of reddish result, which is what we see here. So by day 3 and day 5, we have decreased photosynthetic rates in the control versus the melatonin treatment, which by day 5 is beginning to look a little similar to day 3. And so this was the quantification of this data. And so we can see that our control is this little black line with these black squares, and then melatonin is the blue line with the blue triangles. And so we see by day 3 and day 5, there is a significant difference. That's what these asterisks signify. Significant difference in the chlorophyll content. So the y-axis is chlorophyll content, and the x-axis is time. So we see that, after day 3 and 5, this correlates really well with what we saw up here, day 3 and 5. These have notable yellowing. And so we can quantify that, and that's seen here. And then this ratio of the fluorescence, so the V or FV to FM, this is what we saw in this diagram here. This is that ratio of photosynthetic activity, basically. And this is tan. And so, he says, well, what is ROS homeostasis key to regulating these Chinese flowering cabbage leaf senescence processes? Because that's kind of what the results look like. And so maybe we do an experiment, and results of this work can provide a theoretical basis for extending product shelf life. So this is the part of the experiment now. So they're going to set up this experiment. Now, what they did is they went to this market, which is apparently a popular market, one of the most popular markets, and they bought a bunch of cabbage, this Chinese flowering cabbage from the market. And they applied the melatonin treatment to half of them. So they dipped them in this 100 micromolar solution of melatonin, and then the other half they dipped in distilled water. So they said they went to the market and they bought a bunch that looked most uniform, and then they randomly selected half and half, and then applied this melatonin treatment. They sat in there for like 3 minutes, I think. And so now what we can do is we can, over the course of several days, we can look at these different things like chlorophyll and this FV FM ratio, like they did in the previous experiment from 2019. And so what they did this time was they added a 7th day. So they sampled at 0, 1, 3, 5, and 7 days after applying the melatonin treatment. So they look at the chlorophyll content. So what they did was they shredded the leaf and they extracted it with acetone and ethanol at a 2-to-1 volume ratio. And then they estimated the chlorophyll with a spectrophotometer at these two wavelengths, 663 nanometers and 645 nanometers, because that's the fluorescence peaks for chlorophyll. And then they did this ratio, which is a maximum potential quantum efficiency for photosystem 2. This is kind of like the universal standard for evaluating photosynthetic rate in an extent. So this is a chlorophyll, this is done with a chlorophyll fluorometer. And now what they saw from this was that chlorophyll content in the control decreased more rapidly than the melatonin treatment, which is what they saw in their previous research. But now they have a 7th day here. So by day 3, so on the top we have control, on the bottom we have melatonin. Day 0, day 1, these look pretty similar. By day 3, consistent with the previous research they had done, starts to do some yellowing, a little bit of yellowing here too, but not as significant as day 3, 5, and 7, which are almost wholly yellow at this point. And then this day 7 in melatonin looks kind of like day 3-ish. And so we can then quantify this, and we see, yep, by day 3 and day 5 and day 7, the control, which is our black line, has a very low rate of chlorophyll, and the melatonin treatment in this paper is the red line, which still decreases, but is significantly different and higher in terms of content, chlorophyll content, than that of the control. So melatonin seems to be delaying the rate of degradation of chlorophyll. Still going down, but it's a little slower. So now let's look at this ratio, the FV to FM. So that ratio decreased in the control more rapidly than melatonin treated, as we had seen previously, where by day 7, this whole tip of this leaf isn't even photosynthesizing, really. I mean, it's pow, it's done. And then by day 7, in the melatonin treatment, this looks very similar to maybe like a day 3 or day 5 scenario. Maybe if there were a day 4, that's where it would fit in here. So that's kind of what we expect. And we can quantify this, and consistent with the visual data and from the chlorophyll fluorometer, what we saw with that, with the scale there, by day 3, 5, and 7, we have significant differences. That's what these asterisks mean. So 2 asterisks means very significant. 1 asterisk means statistically significant, but not very significant. And then these little whiskers here, these indicate the variance, or how much the data spreads out. And so if these whiskers overlap, like in day 1, they overlap, this means, well, they're not really significant in terms of the treatment. But now they're pretty different. And this is consistent with what we've seen. Now we can actually look at the hydrogen peroxide, which is H2O2 content. So hydrogen peroxide, as we recall from earlier, is a ROS, reactive oxygen species. And then we can also look at the overall ROS content using a staining method. So this is a histochemical staining. Now what this is the first one to figure out the hydrogen peroxide content is a 3,3-diaminobenzidine staining. It's also called DAB staining. So what this does is it makes the regions of the leaf brown. That indicates where you have your hydrogen peroxide. The other staining we can use is a nitrotetrazolium blue chloride, also called NBT. So NBT staining gives us basic ROS, reactive oxygen species concentration, which are the blue areas. So let's take a look. Now, let's evaluate this. On top as a control, as we have been keeping consistent, and melatonin's on the bottom. So at day 0 and day 1, pretty similar. By day 3, we see some notable browning here. Day 5, both of them have some browning, but this is really concentrated to that area. By day 7, there's a lot more brown stain compared to melatonin, indicating that there's a higher H2O2 content in this leaf. So this is, you basically do some procedures to make this leaf kind of clear, and then you apply your stain. This is called histochemical staining. And so this particular region of the control of day 7 is a lot more hydrogen peroxide rich. Now we can evaluate the overall ROS status. So top is control, bottom is melatonin. Day 0, pretty similar. Day 1, pretty similar. Day 3, starting to look a little bit ROSy or ROS filled. And then by day 5 and day 7, it's significantly stained compared to the melatonin. And so now we want to quantify a couple of these different things. So the first one is called malondialdehyde, also called MDA. I'll call it MDA from now on, because clearly I cannot pronounce this. And this is a marker for oxidative stress. Now what this is we have a ROS that degrades the polyunsaturated lipids, and it makes this MDA. So basically, your ROS start to destroy your membranes, and then MDA comes out, and you can measure the amount of MDA, and that's related to ROS. Now we also want to quantify superoxide, which is an ROS that causes oxidative stress. We kind of talked about this earlier. Additionally, we can look at the H2O2, hydrogen peroxide concentration, which also causes oxidative stress. So, we did the histochemical staining where we kind of looked at the leaves and had a visual qualitative data. Now let's quantify it and get numbers associated with that content. And so let's look at our MDA. Well, the MDA is lower in the melatonin treatment. So the red line is our melatonin treatment, and these open circles with the whiskers here. In day 7, it's really tight. So it looks not like a circle, but kind of like a warning sign or something. But what we see is by day 3, it's significant. By 5, it's significant, very significant. And 7, it's also significant. So it has a lower level of MDA, which indicates that there's less ROS stress, because MDA is a byproduct of the degradation of lipids, polyunsaturated lipids, with ROS. So in the control, it's higher, indicating that there's more ROS stress. So less oxidative stress in our melatonin. Superoxide, well, is lower in the melatonin treatment, which means, there's less ROS in our melatonin treatment. We see here the red line, it's pretty significant throughout. And what about hydrogen peroxide? Well, consistent with our initial photos that we saw, by day 3, that's when we got the big change. So day 3, day 5, and day 7, the hydrogen peroxide, which is an ROS, is lower in melatonin treatment. Now, what we can do next is we can determine the capacity, the scavenging capacity of superoxide, the hydroxyl radical, and DPPH. So what we're doing is we're going to use a kit, and this kit will tell us the capacity to stop these things. So these are the superoxide, hydroxyl radical, and DPPH. These are all ROS stress things. So they all cause oxidative stress. So if we look at superoxide, well, with the kit, some interesting data here, but by the end at day 5 and 7, we have increased scavenging rate. So on the y-axis, this is the amount of superoxide that has been scavenged, which is to say that we've stopped it from doing things. So we basically put it to an end. And so day 1 is significant. Day 3 kind of overlaps here. And we see in the control, it kind of peaks and then goes down. But notably, there's a higher rate of stopping this ROS. Now, if we look at the hydroxyl radical, this is pretty consistent as well. By day 3, 5, and 7, we have an increased rate of scavenging this ROS, this hydroxyl radical. And then this DPPH, also by day 3, 5, and 7, has a higher rate of scavenging, which is to say that we've stopped this reactive oxygen species. Or this one actually has just free radicals on it, but gives us the same kind of data. Now, what we can do is we can actually look at some of these antioxidant enzymes. So let's measure the activity of this, these 3 enzymes. This
It's high noon.
Oh, it's high noon. Okay, we're going to measure the activity of these enzymes, like this peroxidase, which has an EC number of 1.11.17, and I'll talk about these in a minute. But what this does is this peroxidase enzyme, it takes 2 phenolic donors and then our ROS, and then it gives us 2 phenoxyl radicals of the donor and then 2 water molecules. So this is how we stop this ROS here. But we have other ones like SOD, the superoxide dismutase. What this one does is it takes our superoxide and then 2 protons and gives us hydrogen peroxide and oxygen. But now we're making more of this ROS, so we got to get rid of that, right? So then we have a thing called catalase. This enzyme can take 2 of our H2O2 hydrogen peroxide and give us 2 water and 2 oxygen. So what we want to do is we want to measure the activity of these antioxidant enzymes. But before we do that, let's look at this exciting thing called the EC. So I talked about these ECs and we got all these numbers here. The EC is called the enzyme commission number, which is a numerical way to categorize enzymes based on the reactions they catalyze. So basically, whatever they do, we're going to give them a number and we're going to rank them or categorize them so that we can better understand these different enzymatic reactions. Now, Wikipedia has some good tables, and this is one of them. So the first number is based on this general class. Is it an oxidoreductase enzyme, which means it's going to catalyze an oxidation reduction reaction? Or is it a transferase? Or is it a hydroxylase? And so forth. And so what we're looking at are these oxidative reductive reactions where we're trying to stop this highly reactive oxygen species. And so all the enzymes we're looking at are going to be in the EC class 1. So it'll be 1 point something, point something, point something, which just means a different specification. So we're going to be looking at dehydrogenase or oxidase enzymes in this EC1 category. And so we take a look back at our peroxidase, and that's EC1, EC1, EC1. Nice. So let's look at the peroxidase activity. So red is our melatonin treatment. By day 3, 5, and 7, once more, we see that there is an increase in the amount of this peroxidase. So on the y-axis, this is the amount of that enzyme, which is a protein. So the amount of this protein that we've extracted. And sure as sunshine, there's more. SOD, another antioxidant enzyme. By day 1, there's even a notable significant increase in the concentration. So these are antioxidant enzymes that stop ROS. And then we have our catalase enzyme. Kind of weird, maybe, because it doesn't follow the trend that we've been seeing throughout. There's no really significant difference between any of these concentrations. So what's going on with catalase? Well, we'll address that in a bit. All right, so next we can do a quantification of antioxidant enzyme activity and antioxidant levels in this cycle that's called the ascorbate glutathione cycle. This is an important cycle for reducing hydrogen peroxide concentration. So this is a way to stop H2O2. Now, GSH is a reduced glutathione, and ascorbate is reduced as well. And so these two have reducing powers. They can do things. And then our, DHA and GSSG are oxidized. And so here is our glutathione reductase that we've seen, and it takes our oxidized GSSG and reduces it into glutathione, which is a key part of this process to produce ascorbate. And our ascorbate is reduced, and then it's able to quench essentially this H2O2, and then it becomes becomes oxidized into this other form here. And so we can then quantify these, and I'll leave the little diagram down here, and we'll look at each of these one at a time together. So the top one is ascorbate content. We've seen the melatonin treatment. They're pretty similar throughout. At day 3, there's one time point where it's pretty significantly different, but honestly, the concentrations are pretty much the same. Now, if we look at glutathione content in the melatonin treatment, there seems to be a big spike at day 5, and then by day 7 is pretty significant as well. Now, we've seen this one, so this is our ascorbate, and there's not really a difference. So this is where it is in the pathway. Minimal significant difference. Like I said with the glutathione, well, yeah, significant increase here. And so in this part of the pathway, there seems to be a lot in the melatonin treatment. Now, what we can do is we can look at these two. So the DHA and the GSSG, these two are the reduced forms, the oxidized forms of our glutathione and ascorbate. And there seems to be a significant decrease in these with melatonin. So what does this mean? Well, this means that GSH, the glutathione, and ascorbate are being oxidized, which means they're stopping ROS. And so because we have these DHA that are in lower concentrations, then we're led to believe that there might be an increased enzymatic activity. So particularly with DHA, there might be an increase in this restorative enzyme here that brings ascorbate back to our reduced state. So DHAR was probably increased, though that'd be an interesting enzyme to look at, that activity. Additionally, with GSSG concentration or content rather, there is a decrease as well. And so we would assume that something's going on. Maybe one of these things that's going on here that's leading to this significant difference is that we have a glutathione reductase enzyme increase. So this enzyme might be overexpressed, so there's an increase in its production, and it's leading to more glutathione being produced, which is consistent with what we saw at the top here. And so an important thing to note is the ratio between the reduced to oxidized forms. So at this bottom here, with the melatonin treatment, there's a higher ratio of reduced to oxidized in this ascorbate, which leads us to believe that there's a better ability for the melatonin-treated plant to combat H2O2. And also with glutathione and its oxidized form, there's a pretty high ratio here, indicating the same thing. So now we can look at gene expression with quantified, QRT-PCR. So what this is we're basically quantifying the amount of the transcripts. And then we're amplifying it so we can detect it. So these are all of their primers, and they have all these different gene names. So we're going to go through a few of them that are important. We see this one, they all start with BR, which just refers to that genus Brassica and the specific epithet, which is Rapa. So these are the Brassica. These are basically just like homologues of the other genes that they were found in maybe Rhabdopsis or something. So we see that the chlorophyll degradation genes, they decrease with melatonin treatment. So we have relative expression here on our y-axis. Melatonin is in our red, just like the rest of the paper. So these two genes, and control is black. These two genes are PAO and SGR1. Now, these are two genes associated with degrading the chlorophyll. Now, if they increase in relative expression, that means we'd expect less chlorophyll, and we'd expect them to yellow. So consistent with what we saw from our initial visual look, and we saw those yellowing leaves, looks like there's actually more expression of these enzymes that break down chlorophyll. So statistically significant changes, or rather differences. And so a melatonin treatment is lower. Now we can look at the senescence associated genes. These are just a bunch of genes, well, SAG or senescence associated genes are associated with leaf senescence, obviously. So SAG12 is a notable one that seems to be doing a lot of different things. And we see by day 5 and day 7, they actually have to change the scale on the y-axis for the control, and then also the melatonin by day 7. And this expression level kind of skyrockets. So we see that there's a pretty low level of these senescence genes in the melatonin treatment. And even by day 7, it's lower. Even though it increases a lot. SEN4, BR SEN4, also significantly lower throughout. Now we can look at our enzymes that we've been looking at. So this is Nebraska POD, Peroxidase, our Superoxide Dismutase, and Catalase. So we see increases in the melatonin treated plants. And so there's increased POD in all of these, indicating that there's going to be more ROS quenching capacity. Additionally, same thing with the superoxide dismutase, but then catalase is a little bit weird because we have the control by day 3 that's very high, statistically significantly higher, and then by day 5 we have the melatonin, which is significantly higher in its relative expression. So perhaps there is a difference between something is unknown, and we'll address that in a little bit. Now we have this other thing called the respiratory burst oxidase homologues, particularly of it. So there's a bunch of them, like A through E. They showed some decrease in expression with melatonin. So I pulled 2 of these out. So this is the B. So this is, this respiratory burst oxidase homologue B. And we see that there's some decrease by day 3, 7, but day 5 is a little bit weird, but it's not statistically significant. Also in the C form of this, RBOH, day 3, 5, and 7 have significant differences, but by day 7, we have an increase in these genes. So kind of particular. Now, they seem to follow the similar trend of D and D2, but then E is a little bit whack as well. So what does this all mean? Well, let's look at the key points, and let's put everything together and get some useful information. So the first thing is that melatonin significantly reduced the post-harvest leaf senescence in Chinese flowering cabbage. So taking together everything from the chlorophyll data, just visually looking at it, to the activity of photosystem 2, to relative expression of these chlorophyll degradation genes, all this data helps support the finding that this melatonin treatment did reduce the post-harvest leaf senescence, particularly like in the yellowing and the yellowing process. The second thing is that there was an accumulation of this superoxide, this hydrogen peroxide, and MDA content in the post-harvest cabbage leaves. It was considerably reduced by melatonin treatment. So here's our H2O2, hydrogen peroxide. And so we see throughout that the concentration of these free radicals decreased in melatonin treatment here. And then this is the concentration of that actual ROS. And then we also have the enzymatic data that correlates to this, that shows that this peroxidase, that it stops H2O2, was statistically significant at day 3, day 5, and day 7 with increased amounts of that protein. And then we see at day 3, 5, and 7, there's lower amounts of this H2O2, which puts together a nice picture. Now we can then see that the third thing is that melatonin dampened the expression of these respiratory burst oxidase homologues, or the RBOHs. in the cabbage leaves. So RBOHE is believed to be associated with premature senescence, and so that's why it didn't seem to follow a lot of the trends of the other ones like D. So D and D2, they kind of give us this curve that fits all of these pretty similarly. And the melatonin seemed to dampen the expression of these, which cause leaf senescence. The fourth thing is melatonin treatment significantly improved the capacity of post-harvest cabbage leaves for scavenging O2, hydroxyl radical, and the DPPH radicals. So here's our example of DPPH radical scavenging. And we saw that when we applied the melatonin, there was a greater a percent of it scavenged, or stopped rather. Okay, getting near the end. The fifth thing that we took away from this is that melatonin treatment significantly alters the enzyme activity and gene expression of these enzymes. So POD, peroxidase, the SOD, and CAT. So as we talked about, there was an increase in these enzymes, which means that we would assume there'd be increase in ROS quenching or scavenging. With the melatonin treatment, it's significantly higher in POD and SOD. But CAT is a little strange, as we saw. So what's going on? Well, CAT transcription and activity do not correlate. Now, this is common throughout. We don't always see transcription associating with the proteins. They're not always correlated. So if we look at the relative expression and we compare that to the actual protein content, well, not really correlated, but sometimes that's how it works. So perhaps there's some organelle specificity associated with this. Maybe there's involvement of unknown genetic factors between these two steps that helps regulate the catalase protein in the plant. But it's an interesting figure. So CAT might also be CAT might also be due to a delayed response, or might have some delay that we didn't see in the other enzymes. Now, the sixth thing is that there were effects of melatonin treatment on the changes of components in this ascorbate glutathione cycle in post-harvest cabbage leaves. So we saw that this ratio of reduced to oxidize was higher in both. So the melatonin treatment basically gave us a greater ability to have more reducing power to stop the ROS. And then now we can put together a working model of everything we've learned. So if we take into account all the information from this paper, we can determine that if there's a melatonin treatment applied, it will stop the membrane lipid peroxidation, which leads to MDA, which leads to leaf senescence. So we did that KIT test to determine the amount of MDA, and we saw that melatonin stopped this lipid peroxidation. We also saw that melatonin treatment stopped the reactive burst oxygen homologues, which lead to ROS production. And then these ROS fill up in this pool. And then this big pool can then lead to the senescence-associated genes, which lead to the overall leaf senescence. We also saw that we had a favorable amount of ascorbate, glutathione, and then good ratios of the ascorbate to its oxidized form and glutathione to its oxidized form, which promotes a nice, robust ascorbate glutathione cycle, which helps with ROS scavenging, which stops filling up this ROS pool. And so we can kind of decrease this amount of ROS, which would decrease the amount of SAGs expressed, which leads to leaf senescence being decreased. Additionally, we saw that when we have melatonin treatment, there was an increase in the enzyme activity of this APX, the ascorbate peroxidase glutathione reductase, and this other enzyme associated in the cycle, the ascorbate glutathione cycle. And so because of this, we had a better ability to ROS scavenge and stop filling up this pool. We also saw the melatonin treatment increase the activity of our peroxidase and superoxide dismutase enzymes, which led to those proteins being created. These are transcription, so the transcription of these increased, which led to the increase in these proteins, which led to the increase of ROS scavenging, which led to the decrease in the ROS pool. And lastly, we have melatonin treatment stopping the chlorophyll degradation. And so we have a hindrance of chlorophyll degradation with melatonin treatment. And that's kind of how we can take all that information and put it into one picture. See how it relates. So these results may have practical implications for increasing shelf life and commercial product qualities of the important leafy vegetable that is Chinese flowering cabbage. So this was the presentation of this paper. I think it's interesting because, you know, they basically dipped the cabbage into melatonin for like 3 minutes and gave a pretty significant phenotypic change, as well as changes in expression of a lot of enzymes and metabolite changes. And so perhaps this could be used in the market for something. You spray your cabbage in the supermarket with melatonin and it'll stay green longer. Perhaps this is something that we'd be doing in 10, 15, 20 years. Who knows? All right, thank you for listening. Have a good rest of your day.